Today I'm going to talk about a range of applications for the DNB-seq technology, our experiences with it, and I also want to talk about some of the potential future uses that I can see. I've been involved with using molecular markers and genomics of plants for a very long time, writing lots of books over the years about all the developing technologies. But the key factor that we now realize, of course, is that sequencing really has replaced conventional genotyping, that sequencing and genotyping technology, in my mind, converged long ago, and that the most appropriate way to establish a genotype is to sequence it, and the best genotype of an individual is the whole genome sequence. It's much more informative than a part of the genome, and with the current costs and efficiencies that are possible, I'm a great advocate for whole genome sequencing of everything. The technologies that we've used that are related here are the DNB-seq method that has just been well introduced to you. We've also worked with some extensions of that technology or further applications. The STLFR is a method where we effectively create a pseudo long read, and that builds upon the short read technology. And we've done some work comparing the applications of that with Oxford Nanopore and PacBio long read technologies. And the reality, of course, with all of these methods is that they all have strengths and weaknesses and have particular places where they're the right tool. The very exciting and very popular current application of these technologies, of course, is in spatial transcriptomics, the stomics method, where we can beginning to be able to cost effectively do transcriptomics in a spatial way. And that's a very powerful technology because that allows us to look at not only the transcriptome of a tissue or an organ, but to know which component tissues and which cells within that are expressing the genes. And that gives us so much more biological insight, whether we're doing basic research or in fact plant breeding, where we need to know where the genes that we're selecting or breeding for are actually expressed. So the motivations in our work are that we use sequencing as a research tool. My work's evolved a lot in understanding, conserving, particularly the genetic resources of our major crops, and in using that to introduce new traits into agriculture, particularly into food crops, and really the driver to doing that in an accelerated way is the need to adapt to climate change. But I think another very specific area that we do find ourselves applying DNA sequencing and in plant genotyping is in the protection of intellectual property. And in that sense, we need a especially reliable platform because we need something that effectively will stand up to cross-examination in court. We have to have very robust methods for the analysis that we do. We're applying these technologies in breeding selection, gene discovery, really specifically the DNB-seq methods in understanding heterosis in some of the systems by getting a very reliable measure across the whole genome of heterosis, characterizing key genetic resources in actual IP protection cases, where we're really guiding now the conservation of biodiversity with some of this data, and I'll say a little more about that. And we're beginning de novo domestication, where we're using the ability to have a very accurate genome sequence of everything we find to target with CRISPR-Cas methods the quick domestication of completely new species or crop wild relatives to give us new options for agriculture. And our latest effort is a major program to manipulate plant genomes to replace fossil carbon. Here we're trying to replace oil with a renewable plant source for many of the hard to decarbonize applications that we necessarily need to use a plant based carbon source for. So I'll start by giving you sort of what we're doing a little bit in sequencing to characterize wild and domesticated plants. And one of the big programs that we're applying this to is our tree genomics program. We're developing a toolbox of a platform, if you like, of technologies to underpin the production of a lot of horticultural crops. And the latest iteration of that is a project we call Building an Advanced Genomics Platform for Australian Horticulture. These two projects have been funded by Horticulture Innovation at about a $30 million investment in genomics. These are the targets. So we started by building reference genomes for all of these and sequencing the germplasm that's available, and so that we've got a complete genomics basis for breeding of mango, almond, macadamia, avocado, citrus, and in the latest iteration adding pineapple, banana, papaya, custard apple and passion fruit. Now these represent for us the most important, economically most important species, and that's why they're chosen as the targets for developing these genomic platforms. The genomic platform, of course, is key to breeding and genetic improvement, but it's also quite important in production. If we understand the biology of these species better, we can actually better manage their production and manage the quality of the products that we produce from them. So it's not something that's just got application in breeding. It has much broader value for these industries. I like to talk mainly about the macadamia example, which is where we've used a lot of DNBC methods. This is a native species in Australia, one particularly that we've needed to drive. This species was domesticated in Hawaii only about 100 years ago. So, and the domestication is fairly mysterious. Some seeds found their way across to Hawaii. An industry was established. A lot of breeding and selection went on. And then these newly bred varieties were taken back to Australia to establish the industry there. So we've got a very strange situation where a very small genetic base has been the basis of the domesticated gene pool, but we still have a much broader genetic resource in the rainforests in eastern Australia, and we're trying to now establish if we can breed better material by using that broader genetic base. So sequencing everything is the way to understand this system, understand what happened in domestication, understanding what that genetic resource in the wild is. There's 4 species, and so we've targeted collection all of them. They're all now rare to endangered. One of the species is critically endangered. We've got less than 100 individuals. I often think that that's the sort of a threshold. If you can't get to 3 digits, if you've got less than 100 individuals of a species, it's something we need to focus a lot of attention on. In all those cases, of course, we justify sequencing every known individual of the species, and unfortunately there are a growing list of species for which we have sequenced every known individual of the species, beyond just the macadamia example. And so these grow in particular areas in the rainforest, and where these areas unfortunately have been subjected to substantial urbanisation. The city of Brisbane and the Gold Coast occupy much of the former habitat of these species, so there's only very fragmented populations remaining in the wild, and these require urgent support, and genomics is the best way to really understand how we better target that management. The 4 species include 2 with fairly small seeds that are toxic. They have cyanogenic glycosides in them, so they're very bitter. But we need to work with these because they actually are an important resource. Like many horticultural crops, we're trying to change the whole process by making the production much more efficient. The good example, I think, is apples, where we've gone through probably 2 orders of magnitude of improvement in productivity per hectare in the last 100 years or so. For many of the other horticultural crops, they need to go through that same transformation instead of large trees spaced out in the field, we need large populations of small trees close together so that we get much more intense production, reducing the amount of water, nutrient, and other things, and land required to produce these crops. So instead of trees that we have to climb with a ladder to harvest, we need a wall of fruit or a wall of nuts that is presented to a robotic harvester. That's the transition to the future for most horticultural crops. And that's requiring that we understand the genomics of these, but also that we have access to the genetic resources that we need to do that. And these small trees represent the opportunity for that change in architecture that we need of the trees to move them to that much more productive system where we have a much more intense production of closely planted trees. So we've done a lot of work using these actually as a model for studying and improving genome sequencing, assembly, and annotation. In fact, the first study here published in Gigasciences, where we compared all of the available sequencing platforms for sequencing and assembly of genomes using the macadamia as an example. Since we needed to do it, we chose that, and we've subsequently used this same model and iterated it with each version of the technologies that have become available, the improvements in the chemistry in some of the platforms, and of course the also very important improvements in the software tools available for assembly and annotation of genomes. And I have to say, there's been a real revolution in the last couple of years. I think I don't think we can blame COVID, but if you think about what we were doing in genome sequencing before COVID and what we're doing now, it's just a completely different world. To the extent that we're resequencing a lot of things that were sequenced pre-2020, and really I presented yesterday a case where we resequenced rice, and we get 10% more genes with the technology we've got today than we got just a few years ago in something as well known as rice, where we think we knew it all. With increasing technology, we need to go back and revisit, because the greater accuracy of modern sequencing means we can actually find genes that were not clear in the sequence in the past. So it really is a fast-moving field in that regard. And so certainly upgrading the sequencing and the assembly such that with the long-read technologies, we now expect in most cases that each chromosome is a single contig with telomeres on each end. So we don't any longer have fragmented genomes, we have highly accurate genomes. But and it tells us a lot more about it. In the case of macadamia, we now know that about 20 or 30% of the genes are actually duplicated on another chromosome, and that's shown here. And so there was a whole genome duplication in these species, and that is important to understand, because if we're working with markers, that we need to know that maybe 20 or 30% of the genes will be in two places, and that will potentially confound your analysis, unless of course your analysis is accurate enough to be able to distinguish those. And in many cases you will be able to, but only if you've got a method that really is based probably on DNA sequencing. A lot of your conventional chip-based methods just can't really cope with that sort of duplication. And that's probably not uncommon in many of the plants that we work with. And when we think about the 4 species, it starts to become a very complex relationship between the different copies of the same gene across what is effectively one gene pool we have to work with if we're going to breed these trees. So to really go to a significant level of depth with the DNB-seq method, we've focused on 365 wild and domesticated genotypes at about 30x coverage. That's generally the level that we aim for. We nearly always get at least 20x coverage, and sometimes we can get 50. Really, we do need to be in that level for a diploid. Where we're working with polyploids, you need to have, I think, that much coverage per, a tetraploid needs to be twice that if we're really going to analyze it meaningfully. That is sort of the minimum, really. one of the problems that we have in analysis of variants in plants with sequencing is that quite often we've been working with collapsed genomes, and collapsed genomes have one base where when there's a heterozygous position, there's actually two. And DNA sequencing is randomly selecting the sequencing of one of the two alleles. And if you only have a 10x coverage, 1 in a million times, you can do the maths of tossing a coin 10 times in a row, you will actually call falsely a homozygous variant just by chance. So we have to go to that sort of level of sequencing depth with very accurate sequencing to get to avoid those coincidence errors of chance sequencing that are very common, particularly in heterozygous species like these. So when we have this data, there's lots of things we can do with it. One of the things we always do is a nuclear gene phylogeny, where we take 100 or so conserved angiosperm genes and do a phylogeny of the nucleus. But the thing that we get as a bonus always when we have short read sequencing data is the chloroplast genome. Any plant DNA sample that you sequence will give you a chloroplast genome, and it will give you a perfect genome. If you go back into the past, people used to try and extract the chloroplast and sequence it and amplify it by PCR, and they had all sorts of problems because they amplified artifacts, they amplified copies of the chloroplast genome that are inserted in the nucleus. The advantage of the next generation sequencing approach is that you're sampling in a relatively unbiased way all of the DNA that's there. And if you think about a plant cell, you've got one nucleus, but you've got 1,000 chloroplasts. So the frequency of genuine chloroplast sequences compared to those inserted in the nucleus is 1,000 to 1. So if you go with the consensus, you always get the correct sequence of the chloroplast if you sequence the total DNA. If you rely on PCR amplification, there's always a chance your primers selectively amplified an artifactual insertion in the nucleus. And we see that in the literature, in the old literature, very commonly that's what people were seeing because of the biased nature of PCR amplification. So the challenge of getting a perfect chloroplast is completely overcome by next generation sequencing because of that abundance of the chloroplast in your sample. And with the tools we now have to both automatically assemble the chloroplast and to annotate it. So for every sample of plant sample that we've got, even a relatively small amount of sequence data for, we can get a perfect chloroplast. And that's a really useful tool, quite often for identifying a plant, but also for understanding some of the genetics. And what we find, though, is interesting biology. So if you look at these two phylogenies of macadamias, the nuclear genome phylogeny shows that the species are in different colors. It shows 4 very distinct species forming 4 clades on the right. That's exactly what we expect. It's a very it gives us confidence that the 4 species are well identified, that what we think are those 4 species fall into 4 distinct clades. When we look at the chloroplast, we see quite a mess by comparison. What's going on is that chloroplasts are being moved between the species, and that's what we see in almost every plant system that we work with. I can go through long lists of different groups of plants we've studied where we find exactly this, that the chloroplast genome is not representative of the species. It moves. What happens is in the evolution of plants, they diverge, and you see that in the nuclear genome. But there's a low frequency amongst closely related species of hybridization events. They only have to happen very, very rarely, and you get transfer of the chloroplast. The chloroplast is maternally inherited, and once you've inherited, that's what you've got. It doesn't change. So we see in this case that nearly all the species at some point have undergone a hybridization event with one of the others. We don't see any trace of that in the nuclear genome because those hybrids usually backcross with one or the other of the species, but they've picked up the chloroplast, which will maternally be retained. And so we see that and it does say that the chloroplast is not a reliable basis for distinguishing a plant at the species level, but unfortunately we still see the literature full of phylogenies based on chloroplasts, and really this sort of establishes that that's pretty much a nonsense unless you're talking about very big differences. We sometimes even see chloroplasts transfer between genera. These are very rare events, but they're there permanently in the memory of the evolution, because millions of years later we can still see that one rare hybridization event that's moved the chloroplast, so that you've got the lateral transfer in the evolutionary process. So we do learn a lot about, I think, evolutionary biology from having the power of these sorts of tools. So and what we can then do is look at where they are. So we can look at how the genetics relates to location of collection. We can understand where the populations are that are different, what relative effort we need to put into conserving particular populations, because we know which ones are different and which ones are the same. Once you do this sort of analysis, you've got very powerful tools to making all the decisions to manage that effective conservation of the species. So in this case, we're using this to look very much at the heterozygosity of the population. So that tells us about how individual populations, to what extent they're inbreeding or outbreeding, what threat there is to their diversity. We're identifying novel germplasm that hasn't been captured by the breeding programs. We're able to associate genotype and phenotype. We've got phenotypic data on all of this. We've now got the whole genome data. We can actually look at which genes control the traits that are of interest. And we can understand very much better the relationship between the domesticated varieties. We can start to understand what actually happened in Hawaii and what sort of processes have gone through to get to the domesticated varieties we've got today. Some of the other applications that are beyond what we're doing here in the macadamias that we're really pushing in terms of analysis is the deregulation of edited plants. In Australia, as in many other countries, if we use CRISPR systems to knock out a gene, it's not a GMO, but we need to prove that that's the case to satisfy the risk-averse lawyers in our institutions. They want to see the genome sequence to be convinced that we haven't inserted some DNA and that's not actually a GMO before it goes out into the environment. So now, as we're moving to do very large-scale experiments with gene editing, we need large-scale sequencing capacity to be able to effectively deregulate that material and get it out into agricultural production. And equally, we need preemptively for plant breeders to have a DNA fingerprint that can identify their variety, because there's always allegations of people taking other people's varieties or misappropriating them. And the best way to protect your variety is to have the genome sequence. We're also extending this sort of thinking to really threatened species. We've got a threatened species initiative, and increasingly we'll be sequencing not only, as I said, all the species, but every individual of all the species. And this is a good example in relatively newly discovered species, 90-metre tree. It's amazing that we can discover new species that are that big. But we are in the rainforest because they're not easy to distinguish because they're so big and hard to see. But in this case, we're down to 40 individuals. And we're really trying to understand what diversity we've got that we can work with to try and keep this species alive. One of the big questions we're applying the DNA-seq to is understanding the evolution of genome size. In the macadamia family, which is the protea family, and these are a few examples of them, we have 100-fold variation in genome size within a single family of plants. Now, that's an extraordinary range. We expect to see big differences in genome size, but to see that within one plant family is remarkable. And so here we're sequencing species that have got genomes that are 300 megabases up to ones that have got genomes that are 40 gigabases, all within the one family, to try and understand what's going on, what sort of environments the ones with big genomes live in relative to small. It's still, I think, an open question, what's driving that diversity of genome sizes, but we hope by being able to sequence them all, we're going to be able to get some answers to that type of interesting biological question. And we're also working with many other systems. We work with the mango, and in many, and particularly crop wild relatives there. What we've found is that many of the mango species are actually hybrids with domesticated mango. So mangoes have been grown for thousands of years, wild and domesticated together. A lot of the apparent species that taxonomists describe in the wild are probably things that have originated from domesticated hybridization with wild species. We can see that particularly where the chloroplast is telling us that the mother of some of those wild populations was actually a domesticated mango. So some of those insights are proving incredibly valuable in thinking about where we go with mango breeding, which is a big issue for us. We're doing the same thing with citrus, sequencing all the citrus, and we've got some amazing diversity in wild citrus, including resistance to the diseases that are threatening the citrus industry here in the US and elsewhere. Rices, we're working with rices across the whole range in the wild. Amazing diversity. The dots represent different chloroplast types that we find between northern China and northern Australia and across to India, which is the place, the region in which rice was actually domesticated. Some great work needs to be done there because this is really the critical resource for rice food security. And we've got all the species of rice and we're beginning to better understand all of that. And in fact, this was one of the species we're domesticating. So we're editing all the genes that were modified by humans during domestication. And we really do need to be able to sequence first, sequence later, sequence all the time to know exactly what we're doing here. We're going to bring something like this into the food system. We're also going back in time, looking at what plants people were eating in Australia 50,000 years ago, because people were eating harvesting and consuming plants across that landscape, and we don't know what species. So we're out in the deserts now trying to find particularly the cereal relatives that are there, sequencing those, and we've just discovered a new millet that looks very exciting in the deserts in Central Australia. Sequencing's giving us the ability to discover a lot of the things from the past that are so important. So we're about capturing biodiversity in what's now quite a long list of crop wild relatives. We've got about 10% of the diversity of plants in the world in the Australian flora, and we're working through it, trying to mine all those crop relatives. But as I said, we're also using it to support conservation, and the exciting application for me is that now when farmers or land care groups, as we call them in Australia, are working on restoring rainforests and replanting them, we're actually guiding them by using genomics to sequence all of the possible seed sources so that we ensure that when trees are planted in the forest, they retain at least 95% of the allelic diversity that exists in the wild populations. So for the first time, we can give some assurance to conserving biodiversity by using genomics to guide all those restoration ecology projects, which in the past didn't have any basis. They just took seed and really didn't know which ones to plant. Now we can be really deliberate about it, and we're using this to better manage threatened species and to support the appropriate cryopreservation of endangered species as well. And the last sort of application I want to mention is our work in non-food plants. Most plants were domesticated for food. We're now domesticating plants to replace oil, and we probably need completely different plants. We just don't take the food plants and change them. We have to go back to first principles and find the plants that will suit that. And so we've got a major program to do that, which means sequencing lots and lots of plants. These are some of our current targets. Just end by saying that we need low-cost, high-quality genotyping for a whole lot of applications I've talked about. But what we're focusing on is developing the protocols for the various components of that: the sample collection, the DNA extraction, the library preparation, the sequencing, and the data analysis. And for each of those, we need SOPs that are really robust so that we can do large numbers at low cost and have data that's robust and reliable, and in extreme cases will stand up in court. So they're the key issues they're all the people supporting what I've been talking about. Thanks very much.